Continuation of the protein synthesis video, we just learned earlier that modification of released polypeptide chain or polypeptide folding, some other aspects related to the polypeptide formation. This video is about modification of released polypeptide chain or polypeptide folding. The formyl group of the first amino acid, methionine, of the released polypeptide chain is removed by the enzyme deformylase. Some other enzymes like exopeptidases remove some of the amino acids either from N-terminal end or the C-terminal end or from both the ends of the polypeptide chain. Finally, this polypeptide chain alone or along with other chains get folded to assume the tertiary or quaternary structure and it is modified into a functional enzyme. A polypeptide starts folding into its secondary and tertiary structure while it is still being synthesized. The folding is usually spontaneous because the primary sequence of amino acids in polypeptide specifies its three-dimensional structure. Inside the cell, protein folding is aided by a diverse class of proteins called molecular chaperones. Molecular chaperones. They act by inhibiting incorrect molecular interactions and incorrect folding. In fact, chaperone is an English word which means a person, usually an elderly or married woman, who accompanies a young unmarried lady in public as a guide or protector and protects her. That's something which we need to remember to learn this term, chaperones. Just note the spelling, chaperone. Okay. Two of the most widely occurring chaperone families are HSP70 and HSP60. These are heat shock proteins. Number with them represent their molecular weight. HSP70 and HSP60 heat shock proteins. They have a base activity and action to which the role is folding of newly formed polypeptides. The chaperone proteins are also related to protein activity of the cell and to protein transport into cell organelles. Differences in protein synthesis between prokaryotes and eukaryotes. In eukaryotes, most of the genes have introns that separate the actual message for the synthesis of one protein into small coding sequence called exons. With the exception of a few genes in archaebacteria, genes in prokaryotes do not have introns. Then eukaryotic mRNA molecules are monocystonic, containing the coding sequence only for one polypeptide. In prokaryotes, individual bacterial mRNA molecules are polycystonic, having transcript of several genes of a particular metabolic pathway. Eukaryotic mRNA molecules are completely formed before they come out of nucleus into the cytoplasm. In prokaryotes, there is no question of mRNA molecules to move out from the place of origin. In eukaryotes, protein synthesis occurs in the cytoplasm. In prokaryotes, protein synthesis begins even before the transcription of mRNA molecules is completed. This is called coupled transcription translation in case of prokaryotes. The primary mRNA transcript in eukaryotes undergoes processing and splicing to change into a functional mRNA. In prokaryotes, splicing of mRNA transcript does not occur because they doesn't even have introns. Okay, only exons are there, so there is no need for this splicing activity. In eukaryotes, mRNA molecules are modified by the addition of a 5' G cap formed by methylated tyrosine triphosphate. No such cap is formed at 5' end of bacterial mRNA. Bacterial chromosome transcription translation protein cell membrane cell wall inside the cell wall. With eukaryotes, chromosome transcription, intron primary RNA transcript processing mRNA, nuclear pore, nuclear envelope mRNA translation outside of nucleus, then protein, plasma membrane and protein is formed. In eukaryotes, 5' G cap initiates translation by binding the mRNA to small ribosomal subunit, usually at the first codon AUG. In bacteria, translation begins at an AUG codon preceded by a special nucleotide sequence. A poly-A tail formed of about 200 adenine nucleotides is added at the 3' end of mRNA in eukaryotes. Now, poly-A tail is added to bacterial mRNA. In eukaryotes, initiation of polypeptide chain is brought about by special methionine tRNA The methionine tRNA is without formyl group, whereas in prokaryotes, the first or initiation amino acid is formylated methionine, FMET tRNA, FMET. In eukaryotes, the addition of initiation factor is much more than the 3 IFs found in prokaryotes. About 10 IFs have been identified in reticulocytes and red blood cells. They are named by putting E as a prefix to denote their eukaryotic origin. These are EAF1, EF2, EAF3, EAF4, like that. In eukaryotes, mRNA is without ribosome binding sequence as found in prokaryotes. The complex recognizes the 5' cap on 5' end of mRNA. In eukaryotes, small subunit of ribosome gets associated with the initiation amino acid tRNA, metRNA, met, without the help of mRNA. The complex joins mRNA later on. In prokaryotes, 30S subunit, first complex with mRNA, 30S mRNA, which then joins with the FMET tRNA, FMET. Then role of ribosome in protein synthesis. Ribosomes serve as a non-specific workbench for translation of proteins. Single ribosomes, which are not involved in translation, do not occur free in the cytoplasm. Immediately after translation, ribosome dissociate into their large and small subunit. The two join only during translation. The two subunits of ribosomes have the following functions. Large 50S subunit is there, small 30S subunit, E site, P site, A site, large subunit, ribosomal subunit, and mRNA binding site. These are the regions. Some other aspects. Sites of ribosomes for protein synthesis. Bacterial ribosomes have 4 specific sites for protein synthesis. These are mRNA binding site, amino acid site, A site for attachment of incoming amino acid carrying tRNA. This is represented by A site. Peptidyl site, E site for the attachment of tRNA carrying the growing polypeptide gene. Exit site or E site, it is the site from which tRNA leave the ribosome after they have transferred their amino acids to the growing polypeptide. E site is confined to 50S subunits, while A and P sites are present on both 30S and 50S subunits of ribosome. Then role of small ribosomal subunit. The smaller subunit of ribosome, 30S in prokaryotes and 40S in eukaryotes, binds to the first codon of mRNA and forms the initiation complex, which initiates the linking of the first amino acid tRNA, F-methionine tRNA, F-met, to the first codon of mRNA, AUG. The initiation signal of 4 to 5 purine residues on mRNA on 5' end located 8 to 13 base pairs prior to the initiation codon base pair with a complementary pyrimidine-rich sequence near the 3' end of 16S rRNA of 30S ribosomal subunit. Then role of large ribosomal subunit. The large subunit of ribosome provides 2 slots or binding sites for the attachment of 2 tRNA molecules, the peptide tRNA and amino acid tRNA. Another E site is located extensively on large subunit for the exit or release of tRNA after it has performed its function. The large subunit also contains the enzyme peptidyl transferase or peptidyl synthetase, which helps in the formation of peptide bond between the COOH group of amino acid from polypeptide gene and NH2 group of amino acid from amino acid tRNA. This enzyme is a ribozyme. 23S rRNA carries out this function. Then role of tRNA in protein synthesis. The tRNA molecules versus anticodons, we might have learned about this, which read the message expressed in the form of exons, codons in mRNA, and occupy a specific position on mRNA. Each tRNA also recognizes the amino acid that the codon specifies. The tRNA molecules pick up activated amino acid molecules from the cytoplasm, attach it to mRNA at specific codons, and deposit their amino acids to the growing polypeptide gene. The role of mRNA in protein synthesis. mRNA molecule copy the nucleotide sequence from DNA and dictate the sequence of amino acid in the growing polypeptide gene. Then some antibiotics inhibit protein synthesis in bacteria. The list below shows the effect of various antibiotics. Tetracycline inhibits binding of amino acid tRNA to ribosome. Streptomycin inhibits initiation of translation. Neomycin inhibits interaction between tRNA and mRNA. Then chloramphenicol inhibits action of enzyme peptidyl transferase and formation of peptide bonds. Then erythromycin inhibits translocation of ribosome along mRNA. Effect of mutations on protein structure and function. We are going to learn a topic which is not at all related to the earlier one, but mutations, effect of mutations on protein structure and function, a brand new topic. Gene mutations are caused by deletion, addition, and substitution of one or more base pairs in the gene. Such mutations produce change in the structure, composition, or nature of protein produced by the mutant gene. Radiations, UV rays, X rays, gamma rays, or some chemicals cause gene mutations. Sickle cell anemia, thalassemia, phenylketonuria, and albinism, etc., are caused by gene mutations in human beings. So, translocation of protein in case of free ribosomes, terminated protein molecules are released in the cytoplasm and are translocated to mitochondria, chloroplast, and nucleus, or to the Golgi body. So, inside a Golgi, this may undergo glycosylation, forming glycoproteins. In case ribosomes are attributed to ER membrane, the released protein molecules are extruded into the lumen of ER. Some of them become integral part of membranes or are transferred to Golgi complex. In 1975, Kocher Blobel and his colleagues, I'm not sure about the pronunciation, proposed a signal hypothesis for the translocation of proteins from cytoplasm to their destination. According to this hypothesis, each protein molecule has a specific sequence of 15 to 13 amino acids in its N-terminal, which directs the protein to its specific destination. This is called signal sequence or targeting sequence. Signal sequence or targeting sequence across or in, and this process is called protein targeting. Proteins with signal sequence are transferred across or inserted into the membranes of ER. Proteins without signal sequence remain in the cell cytoplasm. So, translocation of protein through ER, that is co-translational transport. Proteins destined for ER are translocated into cisternal space of ER while they are being synthesized on ribosomes attached to rough ER. They have a characteristic signal sequence on N-terminus. Their signal is rich in hydrophobic amino acids. For translocation, their signal sequence is recognized by cytoplasmic receptor particle called signal recognition particle or SRP. Signal recognition particle recognizes an integral membrane protein of ER membrane and helps in binding of polypeptide signal sequence and associated ribosome to the ER membrane. The integral membrane protein is called signal recognition protein receptor or docking protein. The growing polypeptide with its signal sequence is translated to the ER membrane into cisternal space. So SRP stands for signal recognition particle. SRP is a complex of small 7 RNA molecule and 6 protein recognize the signal sequence on the N-terminus of polypeptide. The transport of protein molecules into ER simultaneously with its synthesis is called co-translational transport. Then translocation of proteins to other cell organelles. Protein destined for mitochondria, chloroplast, and nucleus also have specific signal sequences to direct them to their respective organelles. Such proteins are first released from ribosomes into the cytoplasm and then they are transported to respective organelles. Then post-translational processing of proteins. The products of translation are polypeptides. They are biologically inactive. Several steps of folding and biochemical modification are needed. Then only they are able to perform their specific function. Also, most proteins are synthesized in the cytoplasm except those formed in mitochondria and chloroplast. Therefore, they are to be transported to cellular regions where they have to carry out specific biological functions or catalyze specific reactions. First process, binding of initiation factors EIF1A and EIF3 with 40A subunit of ribosome. GTP binds to EIF2. This complex binds to methionine tRNA and this binds to 40S ribosomal subunit forming pre-initiation complex. Then EIF4G binds to the EIF4E cap and through PAB to poly A tail of mRNA it also binds to ELF4A and EIF4B. So 40S ribosomal subunit with bounded methionine tRNA and EIF2 scans mRNA to identify AUG initiation codon. On reaching AUG codon, EIF5 triggers hydrolysis of GTP bound to EIF2. Initiation factors are released and 60 subunits of ribosome binds to 48 subunit. Some other processes will be taking place in this process. And protein folding. The polypeptide chains represent the primary structure. They undergo folding immediately after the chains begin to emerge from the ribosome in the endoplasmic reticulum as a result of folding. The polypeptide chains assume stable three-dimensional secondary and tertiary structures. The secondary structure is achieved by the formation of hydrogen bonds, electrostatic interactions, and disulfide bonds. The overall folding process is described as cooperative because each folding step facilitates and initiates the formation of another favorable interaction. Chaperones bind to amino N-terminal portions of the nascent polypeptide chain and stabilize it in unfolded configuration during the synthesis of polypeptides. The completed polypeptide is then released from the ribosome and is able to fold into its correct three-dimensional conformation. And molecular chaperones and protein folding for simpler, smaller proteins. The protein folding is a spontaneous self-assembly process, but larger proteins assume their native structures with the help of special proteins called molecular chaperones, which help in correct folding and protect the stabilized transitory intermediate. They are also called accessory proteins. They were first discovered in Drosophila and were originally described in heat shock proteins. Now they have been isolated from all prokaryotes and eukaryotes. Molecular chaperones mainly belong to two families, HSP70 and HSP60. We already learned them. HSP in the sense heat shock protein. They act as catalysts and carry out following functions, which are the folding by binding to unfolded or partly folded polypeptide chains, preventing formation of unwanted interaction that do not provide biologically active structure, stabilizing interaction that may otherwise be unstable, avoiding improper or unfaithful hydrophobic interactions, avoid aggregation and precipitation of proteins during their synthesis. Some other aspects like some chaperones couple ATP hydrolysis to protein folding process, bind to folding proteins to hide their exposed hydrophobic amino acid residues. Then biochemical modifications are also there. The biological changes in proteins to achieve their native changes or native functions from our proteolytic cleavage. Proteolytic cleavage, amino acid N-formyl methionine in prokaryotes and methionine in eukaryotes attached to N-terminus of polypeptide chain are removed by hydrolysis. This is called proteolytic cleavage and is an essential step. In approximately 50% of eukaryotic proteins, a acyl group is added to their amino acid end. So amino acid modification, the activity of enzyme and some other proteins is altered by phosphorylation, methylation or hydroxylation of some amino acids. For example, hydroxyl group of serine, threonine and tyrosine are modified by phosphoryl group from ATP. The hydroxylation of proline and lysine changes the two proteins to collagen, a structural protein. Amino acid modification and proteolytic cleavage. An attachment of carbohydrates. In glycoprotein, carbohydrate molecules are covalently bonded to the protein, thus provide immunological protection, cell recognition, and blood clothing. With addition of prosthetic groups, many enzymes can function in association with covalently bound cofactor or prosthetic group. The prosthetic group are hence FAD, biotin, and pantothenic acid. Then trimming of polypeptide chain. In some cases, long polypeptide chains are trimmed by enzyme exopeptidases. The amino acid residues are removed one by one, either by N or C terminus. For example, long polypeptide chain of pediinsulin is cut into a small functional insulin of only 51 amino acids. Then signal molecules are added or removed from the N-terminal end of some proteins. The proteins are synthesized in cytoplasm and are transported to ER, Golgi complex, mitochondria of chloroplasts, or they are excreted out of the cell. Okay. For transportation of their specific destination, proteins have a sequence for up to 30 amino acids at their N-terminal. This is called signal sequence. Once a protein has reached its destination, the signal sequence is removed before it assumes the functional status. Polypeptide chains are also complexed with metal ions or prosthetic group. The tertiary and quaternary level of protein structure is attained only when some metal atom is complexed with it. In iron in case of hemoglobin and Mg in case of chloroplast is essential for their quaternary structure. Once again, biochemical modifications. We just learned about proteolytic cleavage, amino acid modification, attachment of carbohydrates, addition of prosthetic groups, trimming of polypeptide chain, signal molecule, and addition or removal of N-terminal end of some proteins. Then, not signal molecules, signal molecules. Polypeptide chains are also complexed with metal ions or prosthetic group. Thank you for listening.